Hello, my good friends. This is going to be a really interesting, I hope. Well, it's going to be interesting for me because I've been studying the brain and how you think and how your body is constructed and all the bits and pieces. But I got finally to the DNA and what is actually inside your DNA that we don't even know about. And the reason I got into this line of thinking was because of people taking different types of drugs, psychoactive drugs. And they seem to experience very similar types of hallucinations, if you want to call them, or are they opening up some kind of a pathway to some programming that is what I would call a pre-program? All right. Now I'm going to explain this in a little bit of detail. And I have a lot of knowledge on this because I was a software developer, and we had everything was loaded. It was called a retail appliance. It's a computer POS system, point of sale, and all you had to do was put it on a counter, and they just used it. Now, how could that happen? We had everything programmed in hidden programs way in the back. So all they had to do was say, "Are you going to use guest checks for fine dining?" And they click yes, and boop, it becomes enabled. So they're changing one digit, and in the background, it's enabling a huge region of programming that has routines and commands and areas that it saves data in and stores data in and analyzes it and sorts it and does all kind of what we call queries. We worked with SQL, which is it's a database engine that can go out and query all these different areas in these regions and say, okay, what did this do to that, and how did these guys add together, and if you take it from a series of time, what's the all the graphs, it did everything, purchase orders, it did all that stuff. So all that was built in. The guy never had to do anything. Now, what about us? We come we don't come pre-programmed. We come programmable. And that's what we're going to talk about right now, the programming. And all it is, it's called DNA methylation. You say, whoa, that's a big word. Oh, that's scary. No, it's not. It's the simplest thing you ever heard of. It's either this is there on that molecule or it's not there. That's all it is. And what does that mean? In program language, if it's there, it's a 1. If it's not there, it's a 0. That's binary code. And these are only work with the CGs. Now, and you have the TA codes. That's different. We're not worried about those. The CGs are your program codes, and we're going to look into CG islands, which are tons and thousands of these that create the routines that I just talked about, where the guy clicks this one thing, and then it goes out to a CG island, and all that stuff happens, and then all of a sudden you are programmed to be able to do all that other stuff that's out there on that island. Now, that's called junk DNA. I'm not kidding you. I am serious. Here's the problem with science. They make so many assumptions that it's just not meaningless assumptions. They say that DNA is junk if it doesn't code for a protein. What does that mean? DNA, a section of DNA, has a certain code in there that it creates proteins. All right. And the proteins are little molecules. They're all twisted up and everything, and they go out and they do whatever they do. But they do something. They know that. And if they're not there, you're in trouble. And I agree with that. I don't have any problem with that at all. But the other 98.5%, I mean, it's only 1.5% codes. And the other 98.5%, I just junk. Just don't worry about it. No. It's non-coding, and they say it's junk. Well, it's not junk. I mean, that's insanity. Now, they call it the CPG islands. Those are the apps. A kid would say, I want to use this app. And you say, well, click this little spot right here. An app opens up, and sure enough, that's what happens. It goes the one click of this binary digit CG in a section of DNA says, if I'm this way, don't even worry about it. Nothing's going on with it. The guy doesn't use that. He's not up in Alaska. He doesn't have to know anything about cold. If it's just this way, yeah, you better get the guy ready for cold. Whatever it is, I don't. It's program changes, and they can be so subtle that we just have no idea how deep our programming goes. And it seems to be the more you work your program, the easier it gets to make it stretch into all kinds of regions. I mean, that's what happened to me. I had a hard time in the beginning being able to, unwind everything, and now it just seems to unwind right in front of me. I don't have to do anything; it just happens. But anyway, DNA junk, that's your apps. Those are the apps. Let me just show you something. Hold on. Wait a minute, I'll be right back. All right, I talked about the binary digits. Here's all it is. If you have 4-digit code, and you have a 1 in this line, and all the rest are zeros, this means it adds up to be a 1. If you had a 1 in this one, and all the rest are zeros, it's a 2. If you had a 1 here and a 1 way out here, you got an 8 and a 1 is a 9. That's how binary digits work. So 4-bit code, you can go up to 15, well, 16 digits really. It's from 0 to 15. 8-bit code multiplies it up to 256. So you only added more 4 bits, and you go to 256. Well, guess what? CG islands are thousands of digit positions. Now, how they work, I don't know. Are they subroutines and routines built into the same chunk of programming? I don't know. But I do know this, that these sections of programming have what they call promoter codes in the front. And if they're turned on, the rest goes. Now, I don't know if those promoter codes are like, we used to have different levels of what you could do. If you were a certain person, you couldn't get to this level, or, there was all kinds of specialty programming on the inside, coming into the program. What are you allowed to do? Where can you go in that? Can you change things? Can you just look? Can you print things? Can you export things? All of that stuff can be loaded in that first thing, and then you got a ton of stuff that you can do, obviously. That's the way these programs, and that's the way we work. It's exactly the way we work. And when you're born, you don't have these methylation. You cannot methylate. You need these CGs that are unmethylated. This is gone. That's called a methane. CH3 is methane. That's why it's methanylation. All it is either that methane is attached, and I call it a 1 activation code, or it's not there. That's what methylation is. And let me just go here. All right, here's the CG islands. Well, here's the methylation. See, that's what the DNA is. It's all. Well, that's one of the molecules in the DNA. And to program it, you just add a CH3. It's as simple as that. Now, the islands. I had the islands down here somewhere. Hold on, let me get to the CG islands. I was going to mention that I took a course at Johns Hopkins, genomics course, so that I have some knowledge about the genomics I'm talking about. And then I decided to, this morning I just got this from Coursera. This is where they're free. They weren't before. I don't know if they still are. I don't do it anymore. But they just seem to know what you're doing. They sent this to me, recommendations for you, science of the solar system. It's what I'm studying. I don't know how they know this stuff. And, and then you go to anywhere. Look at this. Caltech, Yale, National Research University, University of Colorado, Boulder again, State University of New York, University of Arizona. These are not junk places. University of Copenhagen. I went to University of Geneva for particle physics. I went to, several of them, and, but look at this. You go to any of these, you can just sign up for them, boom, 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 and they'll keep after you, and they'll, once you get into that loop, they take care of you pretty good. They keep an eye on you. Hold on a second. Let's talk about the CG islands. Okay, here's a CG islands. The reason I thought about that, because I'm looking in here and I'm looking at DNA, and I wanted to make a statement that I do have some knowledge about the sequences, and because genomics, that's what it is. Now, this is again from the National Institute, the government agency. Now, this is pretty new stuff. Epigenomics, where there's no junk DNA, and they relate to 80%. They think they've got it figured out now, somewhere around here. Yeah, 80%. Hold on. Let's just read the whole thing, because this is very interesting. Brand new stuff. And it relates exactly to what we're saying, and health. Now, epigenetics refers to functionally relevant modifications of the genome that do not involve a change in the nucleotide sequence. What does that mean? That means that we're modifying the DNA to do different things, but it's this basic structure didn't change. We just added a little molecule here, or one over there, but the base of it didn't change. All right, so examples of such modification are DNA methylation, exactly what I'm talking about, and then it gets histone modification, where they cut the tails off or something. Both modifications serve to regulate gene expression. That's what they're always talking about. Oh, you got a bad gene. You got something like that. It's not expressing correctly. Well, that doesn't, the gene is not the problem. I'll continue on. Both modifications, this methylation, let's just concentrate on DNA methylation. Okay, methylation serves to regulate gene expression without altering the sequences. It's just turning things on and off. Epigenome encodes critical information to regulate these, to express these genes, to turn them on, to make these things happen. Now, the cellular epigenome is established during development and differentiation and maintained during cell division. What does that mean? Cellular epigenome, that's right at the cell level, is established during development and differentiation. So the epigenome it's a tag that says, here's what I'm going to be. I'm going to be a skin cell. I'm going to be an eyeball. I'm going to be a fingernail. And because all the cells are different. They're all different. And once it's methylated to become that cell, it's no more, you can unmethylate it. It's done. That's the kind of cell it's going to be. So it locks on. That's a locked on cell change, because all the cells come out the same. They come out and they say, well, when you're first born, there's just a blob of junk there. There's all a bunch of cells. Nobody knows who they're going to be. And then all of a sudden it establishes topography around what's called a blastula. And they all say, well, I'm over here where an eyeball's supposed to be. I'm over here where an ear is supposed to be, whatever it is. And then they do that methylation, say click, 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 click. That's what you're going to be forever. Now, every time you want to be disposed, some kind of apoptosis comes along, blows you away, we have to replace you, but make sure that you tag that thing to be a cell of the ear or whatever one that you're leaving, because if it's not, you're going to have an eyeball growing on your ear. I mean, it's that kind of thing. It's that simple. It's just basic. What are you going to do? Where are you going to go? Here's how you work. Case closed. Now, let's continue on. So it encodes this critical information to regulate the expression, and then they are locked in at cell division. Now, the instructions are different in each cell. That's what I said. Therefore, the epigenome is cell type specific. So, all the instructions that follow that promoter code are for that specific type of cell. Just like my program that I used to have. Now, nutrient availability, other environmental factors cause changes in other words, when you don't eat right, you're going to screw this whole thing up. Now, recent research suggests that the critical contribution of this area, this epigenome, to the development of complex gene environmental diseases, including chronic, well, they go kidney disease, any chronic disease, anyone, every single one of them. And it is related to this specific epigenome process that they're talking about. In my world, I call that the probiome. It's the area in your whole body that promotes you to be healthy biologically. All right, so these are bacteria and things that are creating enzymes to help you. Now, they may not understand that at all here. They're talking about the cells have to be certain ways and work, yes. But to turn on and off, they need to be talked to by a bacteria. And you say, oh, Roger, how could you possibly say that? Well, here's how I can say it, because, well, let's go look at why I can say it. Well, before that, let's just finish up with this. I always seem to get ahead of myself. Now, so what is epigenetics? It means the above genetics. It's the study of gene expression regulating that cannot be directly attributed to changes in the DNA. Among the 3 billion nucleotides, less than 2% are responsible for coding. Remember I said before 1.5 are coding proteins, coding DNA to make proteins. The rest of the sequences are long thought to be non-functional junk, possibly only there as a buffering zone for mutations. Yeah, they just threw them in there. Recent reports, especially those published by ENCODE Consortium, indicate that by describing and annotating the cellular epigenomics functional roles should possibly be assigned to as much as 80% of that junk. It seems that this instruction manual that governs gene expression is hidden somewhere in the junk DNA. Exactly. Those are the enable codes that go out into that junk DNA, into that CG island, and say, okay, enable that island to work with all of the bacteria. Really, it's what they do is work with bacteria. And that's what I'm thinking. And it worked with the bacteria through water. Water is the thing that it is that's the intelligence, and that is the source of life. And I'm going to tell you right now, all of this research I've done has brought me to God, and God's Spirit was on the face of the waters. I remember that. Now, God, because nobody there is absolutely no functioning human mind that can accept the premise that everything that we see and are capable of and do and just happened by accident from something exploding, which was from nothing, and then every elegant little thing happened after that just accidentally. That is the weakest mind that has ever. God would be disgraced if he knew this was happening. All right, now, so let's come down to where we got, all this stuff is nonsense now. So we're down to the point where we are a programmable, functioning creature that is elegantly designed by God, as far as I'm concerned. Take it anywhere you want. Now, the epigenome involves marking chemical modification of DNA associated proteins. The marks include cytosine modifications. That's the C. Mainly methylation. We're turning the C on. All right? Methylation, mainly methylation of the cytosine. That's what I've been saying. Then they got the histone tail modification. Forget that. The epigenome is inherited during cell division to maintain cell identity. In other words, that cell has to be exactly like the cell that it divided from. If it's not, you got an issue. You're going to have some kind of mutation. So it has to come from the same cell. However, nutrition and other environmental factors change these tags. Therefore, these chemical tags serve as the intersection between stability and changing environment. All right, so they're normally just everything's cool unless you come invade me and I'm going to turn on and go after you. No. But everything's stable. No, I don't have any problems right this moment, but that'll change quick, I'm sure. Now, characterization of the epigenome helped tremendously to define cell type specific gene expressions. A typical transcription unit in a multicellular eukaryote contains both clusters of proximal promoter, remember that? The proximal promoter genes, elements, and 5 types of cis-acting regulatory sequences. Cis is the cytosine. Now, insulators, promoters, enhancers, silencers, these are program routines The insulator areas border and separate transition units. The promoter is usually located at the 5' end of the transcription start site. And it says, oh, here's how it's going to work. And then they contain elements called TATA boxes, which is bound by either a TATA binding protein or a cluster of cysteine, guanine, nucleotide pairs, CGs. Right? These are the CG islands within this linear sequence known as the C. The P is a phosphate group. It's always in between. You don't even have to think about it. So the CG island, I'm sorry, let's go back to that again. The TATA binding proteins or a cluster of cysteine, cytosine, guanine, nucleotide pairs within the linear sequence. So that's the sequence. They are known as CPG islands. They target gene transcription to take place not only promoters but as long and short range regulatory regions. Regions are needed. These cis-type gene regulatory regions are highly cell type specific. Cell type specific. They are their regions, they're islands that they don't know what they do, but they're specific to certain cells. They're critical for cell type specific transcription. Simultaneous binding of transcription factors to each other to the long end and short range regulatory regions results in genomic DNA loops and join distant regulatory DNA sequences together. That's just gobbledygook. Don't worry about that. Whoops. Oh boy. All right, these are CPG islands. No longer junk. There is no more junk. This is from the National Institute of Health. This talks about modifications, mainly DNA methylation. Right? DNA methylation, the CG turning on and off, is what does all of this programming. Right? So they decided, they realized this after a while, that it's very cell specific, it's all these specific things that they never really thought about, and they just thought it was junk. Well, it's not. So then he decided we're going to think about epigenetics. And epigenetics means epi, above genetics. So we know that there's genetics there. We know all these numbers are there. We just don't know what they do. So we have to come up here and look down and see what do these things do. Well, all this junk DNA, remember I said 1.5%. Well, they said less than 2% responsible for coding proteins. That's okay. But the rest still does something. Now, they just started to look in deeper, and they say now if they can assign as much as 80% of the genome, it seems that the instruction manual that governs the gene expression is hidden inside the junk DNA, which is these promoter codes and all these codes that are in there, and they come down here, and it shows that they've found where there's the promoter codes in here, transcriptions, and in other words, there's a whole chunk of stuff, a bunch of stuff that does something, but you still need a lot of prep information as to how you're going to get into that, and who's going to get into it, and all those different things. I think I already talked about that. But this is where these islands occur, and they're gigantic islands that were sitting out there. Well, 98.5% of your DNA, they just had no idea about. But this is pretty new, and it's all about chronic disease. They're talking about chronic kidney disease. Chronic kidney disease is just one chronic disease. 100% in my world of chronic illness is due to either you don't have the bacteria in your body that was supposed to be there in these certain places, so they are becoming invaded by other things that the mucus should have kept them away, or the enzyme that it made it should have created an environment that was safe. Whatever happened, you're being invaded in that spot, all right, because of bacteria. The bacteria is not there. And then what will happen is you'll be invaded on top of that area by something that shouldn't be there and now takes up residence there. And then you're really in trouble because you can't get it out of there to repopulate it with what you were hoping to get it out. Now, they have been doing, just because we're in this region, now I'm going to talk about something that we've been doing for the last year or so, is autism, and looking into the gut destruction that causes autism. And it does. And the top researchers now understand this, and they're just a little slow on moving forward on it. Now, that's the type of problem that happens in academia, and scholars and experts, they don't want to they don't want to say anything until they can say everything. And then when they say everything, it's so confusing that it's not right 99%, well, it's so far 100% of the time. Now, the only one I'm seeing right now, and I have been in communication with her, is Stephanie Seneff from MIT. And she's working on the glyphosate problem with Roundup. And glyphosate is a bacteria, a herbicide that kills, weeds and so forth. Now, they create plants, corn and all that business, that can tolerate it. They're changing the codes, and they're turning CGs on and off inside the corn to say, don't worry about this other molecule. When it comes, from now on, this area won't be able to attach to it, so it can't hurt you. And then they just sell those seeds. And, Monsanto, I think, or whoever it is that's doing this. But and then they put the Roundup over, just spray it on like and it doesn't kill the crop, it kills everything else. So they think they're in good shape. Well, it does a bunch of other things too. It is partially incorporated into that plant that you eat, is what Dr. Seneff is saying. And, you see the chemistry. You want to see the paper she sent me? I'll show it to you. Blow your mind out of your head. She's like, I sent you a little note about it. All right, but anyway, the chemistry is not well understood by anybody, probably outside of her. However, it is, in my thinking, it's the one that kills your lymph nodes, is possibly from the glyphosate. I don't know. I don't want to step in her area, but I do know that anytime you kill the bacteria in the body, antibiotics, MMR vaccines, glyphosate, any of that stuff, you are going to be chronically ill. You're going to have a chronic ill condition. And then that, it goes for everything. Your skin, it can bother your hair, go for your lymph nodes. It depends on the bacteria. And then you might be specific for one region in your body that's infected with lymph nodes. It might be just under your arms, because that specific bacteria that's supposed to affect, that specific sweat that's coming out of there is not there to deal with it. So you get these invasions happening. Before long, they break through the walls of your body into the vital areas, and you're really in trouble. So, I know I'm throwing medicine into DNA, but it's all, we are not separable from anything. There is not anything that we are separable from, the entire universe, really. I got a lot of research on that too, going out into space and seeing what's out there, and looking at that stuff. It's very interesting there as well. But let's stay on Earth for the time being.